Welcome everyone. Today we are going to learn about cholesterol in more details. In my previous video I have mentioned that plasma membrane contains three major types of lipid molecules. One is phosphoglycerides, second is sphingolipids, and third is cholesterol. In my previous video I have already discussed about phosphoglycerides and sphingolipids in more details. Now today we are going to learn about cholesterol. Okay. So at first we have to remember that, as I have mentioned, the backbone of phosphoglyceride lipid molecules is glycerol. The backbone for sphingolipids is sphingosine, and in case of cholesterol the backbone is actually a four-ring isoprenoid base, based hydrocarbon molecule. Another important point, so this is the backbone. I'm writing here, this is the backbone of this molecule. Now this cholesterol, as I have already mentioned, that all the lipids present in the plasma membrane are amphipathic in nature. So cholesterol is also amphipathic in nature. Amphipathic or amphiphilic, you can remember any term. What does it mean amphipathic? When any structure, basically biomolecules, contain both the hydrophilic and hydrophobic portion, that means water fearing and water loving portion, we call amphipathic. Let's see the structure of cholesterol. Let's suppose this is the rigid steroid ring, that means this is the isoprenoid ring. Let's suppose this is the 1, 2, 3 and 4 isoprenoid ring, rigid steroid ring. In case of cholesterol, one hydroxyl group is actually present that acts as a polar or hydrophilic portion of the molecule. And in case of cholesterol also, there is a short hydrocarbon chain. So this is the short hydrocarbon chain, and these two structure, basically these two structure, this rigid steroid ring and the short hydrocarbon tail togetherly make the hydrophobic portion of the molecule, hydrophobic. Okay, so that's why it is amphipathic in nature. Now third point we can remember about cholesterol, that it is absent, cholesterol is absent in most prokaryotes and absent in all plant cell plasma membrane. Remember this, in all plant cell plasma membrane cholesterol is not present, and in many prokaryotes it is absent. Another point we can remember about cholesterol is that this cholesterol is actually evenly distributed. This is important point, remember this. This cholesterol is evenly distributed between the two layers of the plasma membrane. What does it mean? I have mentioned in my previous video that plasma membrane, on basic facts of plasma membrane contains two lipid layers, so it is called lipid bilayer. Let's suppose this is the cytosol, let's suppose this is the cytosol or cytoplasm. So in case of plasma membrane there is two lipid layers. The outer layer, you can say it is outer inner, but in scientific nomenclature we call the outer layer exoplasmic leaflet, and the inner layer of the plasma membrane is called, the lipid layer is called cytosolic leaflet. Why it is called cytosolic leaflet? Because this lipid layer actually faces the cytoplasm, and as this layer actually is outward towards the cytoplasm, this is called exo means outward towards the cytoplasm. That's why this is called exoplasmic leaflet. Now in case of cholesterol, Cholesterols are evenly distributed among the two layer of the between the two layer of plasma membrane. Why it is important? Because the other two lipids like phosphoglycerides and sphingolipids are not evenly distributed. What is evenly distributed? That means the cholesterol can be present both the inner and outer layer. Okay, it will present both the exoplasmic lipid and cytosolic lipid. But in case of phosphoglycerides and sphingolipids, they are not evenly distributed. They call asymmetric distribution. They are asymmetrically distributed. The phosphoglycerides asymmetrically distributed. So phosphoglycerides and sphingolipids, these two lipids are asymmetrically distributed, and this asymmetrical distribution plays an important role in the plasma membrane. I will take a separate video on this topic, asymmetrical distribution of phosphoglycerides and sphingolipids, but cholesterol is evenly distributed. Now, another point we can remember about this, that when cholesterol mixes with phospholipids, obviously cholesterol is not a phospholipid, so I can write that it is not a phospholipid. In my classification of lipid molecules in the plasma membrane, I have mentioned that phospholipids are those lipids present in the plasma membrane that contain phosphate group, but cholesterol does not contain any phosphate group. That is why it is not a phospholipid. Now, 6th point, we can remember that when this cholesterol mixed with these phosphoglycerides and sphingolipids, it actually enhances or increases the permeability barrier. I have mentioned also in the basic concepts of plasma membrane that plasma membrane acts as a permeability barrier. What does it mean? Permeability barrier means it does not allow most of the water soluble molecules to enter without any assistance. Means this lipid bilayer of the plasma membrane actually prevent polar molecules from inside to get entry into the inside of the cell or outside of the cells, because as the plasma membrane is lipid in nature, so it will prevent, lipid means non-polar in nature, so they will prevent polar molecules. So water soluble molecules like glucose, fructose, they are actually, or even any charged particles are actually prevented through the lipid barriers. So for the transportation of molecules that are not actually come through the lipid barriers, there are transport proteins are present, or channels are present for the ions and other molecules. So it enhances the permeability barrier. So basically the plasma membrane, the lipid bilayer, has a permeability barrier, so it can actually prevent the permeability of many molecules. That's why plasma membrane is also known as semi-permeable membrane. Some molecules are actually permeable, but many molecules are not permeable. So when cholesterols are actually mixed with phosphoglycerides and sphingolipids, it enhances the permeability barrier. That means it actually prevents more molecules to enter inside the cells or actually transport through the plasma membrane. This is another important point. It is really important in the cholesterol. Next point is it actually the cholesterol can perform flip-flop movement. Now, what is flip-flop? I will also explain. It can perform flip-flop movement very rapidly. Okay, in a time scale of just seconds. And here is the difference between this cholesterol and phosphoglycerides and sphingolipids, because very rarely these phosphoglycerides and sphingolipids are seen to perform flip-flop movement. Now, what is flip-flop movement? Flip-flop means, let's suppose any molecules or any structure is present in the exoplasmic leaflet. If it comes to the inside or cytosolic leaflet, or from cytosolic leaflet to the outside leaflet, this is called flip-flop movement. Cholesterol can flip-flop from one leaflet to the other very rapidly on a time scale of seconds. But phosphoglycerides and sphingolipids, here is a difference. They rarely perform flip-flop movement, but perform on a very longer periods. It takes about hours, basically 24 hours or more to perform flip-flop. So here is another difference between phosphoglycerides and here also the between the cholesterol. So it is evenly distributed, it is asymmetrically distributed. Now the point 8 we can remember is that cholesterol actually maintain proper fluidity of the membrane, because membrane is not rigid in structure. I have already mentioned in my basic facts of the on plasma membrane. Membrane is not rigid structure, it can bend and flex. So cholesterol can actually maintain proper fluidity as well as rigidity. Okay, this is very important point. I will take a separate video or class to explain how cholesterol actually maintain the fluidity and also the rigidity. Now another point we can remember, these are all important points for cholesterol molecules. Another important point is that cholesterol increases the thickness of bilayer. Okay, it can increase the thickness of the bilayer. How it occurs, let me explain. Let's suppose this is a phosphatidylcholine. Let's suppose this is a phosphatidylcholine. Let me draw the fatty acid tail in red. Let's suppose this is a phosphatidylcholine and this is the bilayer. What does it mean? Bilayer means this is the lipid bilayer. So if this is a phosphatidylcholine in under the phosphoglycerides, phosphatidylcholine is a lipid molecules under the phosphoglycerides. Let's suppose this is phosphatidylcholine, then the thickness of the phosphatidylcholine if present in the both bilayer is approximately 3.5 nanometer. Okay, now when cholesterol actually intercalates between with this phosphatidylcholine, then the thickness increases up to 4 nanometer. So it actually increases the thickness. Okay, I'm not showing, but just you remember cholesterol when intercalates, the term is intercalate, okay, with the phosphatidylcholines or phosphoglycerides, it actually increases the thickness. But here is a restrictions. Why? Because sphingolipids, let's suppose this is a sphingolipids or sphingomyelin. Let's suppose in case of sphingomyelin, I have already mentioned what is sphingomyelin. If you have not watched previous my video, you can at first watch what is sphingomyelin. Let's suppose the sphingomyelin is a lipid that falls under the sphingolipids. Okay, so sphingomyelin also has a head group and a tail group, but the tail group of the sphingomyelin is actually more longer than the phosphoglycerides. So if two sphingomyelin present in the bilayer, let's suppose here, if two bilayer is present contain the sphingomyelin, then actually the sphingomyelin fatty acid is more longer than the phosphoglycerides. I have already discussed about that. The length of the fatty acids, the number of carbon molecules is more than the phosphatidylcholine or other phosphoglycerides. So it is more stable or more ordered, you can say, more stable. So it is the example of sphingomyelin. It is more stable or ordered. I have already discussed about this that the fatty acids present in the sphingomyelin or sphingolipids are more longer and more saturated. I have already discussed that the saturated, more the fatty acids are saturated, the fatty acids become straight. A fatty acid which with no carbon-carbon double bonds is more straight. If any fatty acids contain unsaturated fatty acids, that means CC carbon carbon double bond, then the fatty acid becomes so kink, it bends the structure. Okay, so in case of sphingomyelin, the thickness of the both sphingomyelin is large, 4.6 to 5.6 nanometer. But when cholesterol intercalates with the sphingomyelin, let's suppose cholesterol intercalates with the sphingomyelin, then it will not increase the thickness of the bilayer. So it is only restricted for phosphoglycerides, not with sphingolipids, and I have mentioned the reason, because the fatty acid chain length is actually longer and more saturated. I have already discussed in my phospholipids video. So it increases the thickness of the bilayer, but remember the condition is it should be a phosphoglycerides. The last point we have to remember about the cholesterol, importance of the cholesterol molecules in the plasma membrane, cholesterol maintains the integrity of lipid rafts. Now what is lipid rafts? I will explain in another video. Lipid rafts are basically some domains in the plasma membrane, and this cholesterol actually helps to maintain the integrity of the lipid rafts. Now integrity means it has been found, I will actually discuss about this in my lipid rafts video. It has been found that if you collect all the cholesterol, if you extract all the cholesterol from the lipid rafts, it will destroy. It will not actually remain as it looks when cholesterol is present. So it will disintegrate. Hope this video will help you. Thank you for watching, and always keep learning.